I'd like to turn it over to Peter.
Thank you, Sarah, for that nice introduction. Are you able to hear me okay?
I can. Thank you, Peter.
All right. Thank you all for joining us today for our talk on guidelines for sizing experimental mouse colonies. So do you find that the production of your experimental mouse colonies is not matching your research needs? Maybe you're unsure of how many breeders to set up to produce the numbers of mice that you need for your studies. Maybe you're estimating animal costs for a grant, or you're trying to plan the expected number of animals needed for your animal care and use protocol. So in this presentation, I'm going to provide you with a detailed guide on how to accurately size your experimental mouse colonies to provide the mice you need when you need them using a minimum of resources. Now, before I get into that, I will take just a couple minutes to introduce you to the Jackson Laboratory for those of you who may not be that familiar with who we are and what we do. So we are an independent nonprofit research institution. Our mission is to discover precise genomic solutions for disease and empower the global biomedical community, in which you're all part of, in our shared quest to improve human health. And we advance our missions in three ways. First is we do have research staff with their own funding needs, like NIH grants, and they investigate the genetics and biology of human disease, mostly using the laboratory mouse as a research organism. We also offer a whole host of scientific services and resources to investigators around the world. In addition to our clinical and research services that some of you may already be familiar with, we also have a host of freely available resources that I will touch on in more detail shortly. And then finally, we're very committed to educational outreach. In addition to webinars such as the one that you're listening to, so the Jackson Laboratory, we are the world's largest repository of mice. We are funded by the National Institutes of Health in the United States to import as many strains as we can and make them available to researchers around the world. We have more than 11,000 mouse strains that are available, and we typically import somewhere between 500 and 700 strains every single year, and we ship millions of mice to researchers around the world. Mice from the Jackson Laboratory have unsurpassed genetic quality and animal health, as well as we also offer comprehensive phenotypic descriptions and references of our strains on our website. We are home to the common inbred strains like C57 Black 6J and Balb CJ, and it's the use of those mice that supports the development and collection of all the other specialty strains, as well as all of the other research resources that we make available. And these do include, starts with our JAX Mice database, which is our online catalog where you can search for mouse strains, but also find information on reported phenotypes and references of other people who have used those mice. We also maintain the Mouse Genome Informatics database, which is a bioinformatics database where we have scientific curators who read published literature on mouse genes and mutations and transgenes and then curate that into a searchable format. And it is not limited to strains available from the Jackson Laboratory. So it's a really great way to find what mouse strains may be available in the published literature to help you with your search, finding your right strains. We also maintain the Mouse Phenome database, which is an international collaborative effort to collect baseline phenotypic data for the commonly used inbred mouse strains that are available from the Jackson Laboratory, so that you don't have to spend your time and resources to figure out those baselines, but you can then get your research done a little bit more quickly. And we also maintain, again, the Mouse Tumor Biology database, which is a scientific curated database to look for mouse models of cancer. And the JAX Clinical Knowledge Base, which is for oncologists and clinicians looking at mutations and of human cancer to help with diagnostics. Okay, so just to give you an idea of what we're going to talk about today, we're going to start with breeding performance data collection, which is the heart of being able to size your colonies appropriately. We'll talk about defining your research needs, and then calculating the number of breeding females that you need to maintain a steady state colony. And then we'll talk about how the decisions that you make in defining what your research needs are impact colony size so that you can adjust your research needs to match your existing resources. So before I launch into that, I just want to review some of the basic reproductive characteristics of laboratory mice. Gestation in mice is about 18.5 to 21 days. There can be variation between different strains, such as C57 Black 6 tends to be about 18.5. Balb C is about 20 days. AJ is about 21. Litter sizes range anywhere to 1 to 2 pups for poor breeding litters to 12 or more for strains that breed really well. And then weaning age for most strains, we typically wean at 3 weeks of age or 21 days. But if we know that maybe at 3 weeks of age the mice are a little small or a little bit more fragile, we may give them an extra week to put on some more weight and just get a little bit older before we might wean them. But that will be strain dependent. And most mice reach sexual maturity at about 5 to 8 weeks of age. Females tend to be a little bit earlier at 5 to 6 weeks, males a little bit later at 6 to 8 weeks. And the most productive breeding lifespan of most strains is about a 7 to 8 month period. And what I want to draw your attention to is that if you look at this sort of timing here, you're looking at 3 weeks for gestation, 3 weeks till they wean, and then another 3 weeks until they reach sexual maturity, which means you're looking at a minimum of 9 weeks every generation. That's assuming the mice breed the day you put them in, which doesn't always happen. So a really good rule of thumb is about 3 months for every generation. And since a lot of us are working from home and not able to work with our mice, as you're starting to think about getting back into the lab and where your projects are going to be, if you need to start expanding your colony or do a breeding like a Crelox breeding, now might be the time to start thinking about getting that started. Jackson Laboratory, we are fully operational and we can take on breeding projects and maybe help you get a couple rounds of breeding done so that when you get back into the lab, you don't have to spend time preparing. The mice might be much closer to readily available so that when you get in, you can hit the ground running and get your studies underway. Okay, so the heart of being able to size your colonies correctly is good data collection and record keeping. It's absolutely critical for successfully managing your colonies as efficiently as possible. It's going to allow you to improve your efficiency so that you're using a minimum of resources, and then also will detect any problems or mutations that might arise in your colony early so that you can deal with it earlier before it really disrupts their colony and your research. And the heart of that is your pedigree book, or whatever tool you're using to monitor the breeding performance of your colony. But the statistics that we use to gauge the breeding performance of a colony is the weaned pups per female per week. And that's calculated, you need to know 3 things. You need to know the average number of pups weaned per litter, and you calculate that by taking the total number of pups weaned, and you divide it by the total number of litters. You also need to know the average number of litters per female, and you take the total number of litters and you divide that by the total number of females. But you have to be sure to include any non-productive females. So any females that don't produce any litters, you have to include them in this total number of females. Otherwise, you will underestimate the total number of females in your colony, and you'll overestimate the breeding performance, and you'll underperform. And then you also have to know what the breeding rotation. That's the length of time between the day you set up your breeders to the day that you retire them and you pull them out of your breeding rotation. If you're not using a set rotation, 30 weeks or 36 weeks or whatever it is, what you want to do is look for when the litter sizes and or the frequency of your litters begins to decline. And it's at that point that's where you want to retire breeders. I'll talk about that in just a little bit as well. You want to calculate your genotype ratio if you have a segregating colony. So for example, if you're breeding a heterozygote to a heterozygote, you need to know, are you getting 25% homozygotes? Or are you only getting 20% homozygotes? Because that's going to affect how you size your colony. You want to monitor the sex ratio to make sure that you're getting close to even males and females. And you want to evaluate this data regularly, at least every 6 months, so that you can then make any adjustments you need in your colony or investigate any issues that arise. And just in case maybe right now you don't have the data that you need to calculate the wean per female per week, this table here may be able to help you. This is just some breeding productivity ranges for common inbred backgrounds that we collected from a variety of inbred, mutant, transgenic mouse strains on these backgrounds, and giving you a range that you can potentially use until you have collected the appropriate data. But I want you to realize that each of these, even though they're on the same background, depending on whether they have a mutation or transgene or they're inbred, there is a significant range, which is why you need to understand how well your mice breed in your facility to accurately size your colonies. Just some general assumptions about what I'm going to talk about today. So I am really going to be talking about an existing colony. I'm not going to talk about how you build a new colony from a limited number of breeders. I'm also going to assume that you have mixed age breeders, and again, I'll be talking about the importance of that. And then for most of the numbers I'm using, cover with breeding in pairs or trios. Here at the Jackson Laboratory, we rarely use harem matings where you have one male with more than 2 females, albeit as long as you're calculating the number of breeding females you need, you should be able to easily adapt to using harems in terms of how large or how many boxes you need on the shelf. And then also we're assuming close to even sex ratios. Few strains really show significant skewings, but if your strain does, that is something, an adjustment you may need to make then in how you size your colony to adjust for that. And then what I also want to remind everybody is that we're dealing with estimates and based on averages, but breeding is a biological process. It can be affected by a whole host of environmental factors. And so just keep in mind that we're trying to estimate a biological process. And it's not always going to be completely perfect every time, but we're doing our best to estimate it as best as possible. So if you look at an individual breeding females, as they get older, the breeding productivity changes over time. So with new females sometimes are not as productive, and then they hit this optimal breeding window, and then as they get older, the breeding performance begin to decline. The age of decline is usually about 7 to 9 months of age for most strains, but you really want to look at the breeding performance of your strain to figure out when the end of that productive breeding window is. And the reason you want to know that is because what you want to do is have a mixed age colony where you have mice at different ages through your colony. And the reason you want to do that is if all your breeders are the same age, what's going to happen is the breeding performance is going to increase and then it's going to decrease. Then you replace your breeders and it's going to increase and decrease. And what's going to happen is you have inconsistent breeding performance over that colony. But if you imagine every female you're starting a week apart or a month apart, you're going to have multiple of these curves overlapping. And what that's actually going to do is actually give you a curve for the whole colony that's much flatter, which means the breeding performance with a mixed age colony is going to be more consistent over time. And if the breeding performance of your colony is more consistent, it becomes more predictable, and that you're going to know when your mice are available to do your experiments, and you can plan those experiments a lot better and manage your time better if you have a mixed age colony. All right, so to determine how large your colony needs to be, you need to make a number of decisions. You need to know how many mice you need and whether you can take less. You need to know the genotypes of the mice you need. Do you need homozygotes, heterozygotes, or hemizygotes? Do you need males or females or both sexes? And then what kind of age range of mice you need, how frequently you need them, what your breeding scheme is going to be. You're breeding home by home to get 100% homes, breeding het by het to get, assuming 25% homozygotes, et cetera. And then also what your controls are. Are you getting them from the same colony, or are you getting from another strain or another colony? So what I'm going to do is just use this as an example as we talk about how to size your colony, just using a standard knockout that's on a congenic C57 Black 6 background. So we're talking about a knockout for gene X. Experimental mice are going to be homozygous for that gene X knockout. Those homozygotes are viable and fertile, and we can breed homozygous knockout to homozygous knockout. Control mice are going to be C57 Black 6 mice, which are wild type for gene X. These could be that something you buy from a vendor of Black 6 mice when you need them, or you could have your own Black 6 colony. But I'm only going to focus right now on the experimental mice. If you have your own colony of control mice, you're going to do the exact same thing. You would just have another colony of those mice for your controls. But we're just going to focus right now on the experimental mice, the homozygotes for that gene X knockout. So in this case, what we're going to start is with where we want 20 mixed-sex homozygotes at a 5 to 8 weeks of age. That's a 4-week age range. And we're going to look at getting that cohort of mice every month. So every 4 weeks, we need those 20 mixed-sex homozygotes. And then the breeding performance I'm going to use for every example so that it's always consistent so that we can see how when we change some of these parameters, how that affects the colony size. I'm going to assume that they're using an average number of pups weaned per litter is 5. The average number of litters per female is 2.8. And the breeding rotation is 26 weeks. These really don't correspond to any mouse strain that we have. I just use them to work with the examples today. And ultimately, as you'll see, I went with the low end of the C57 Black 6J wean per female per week range. Okay, so the steps are, and if you were able to download or have that worksheet, I'm basically going to start going through that worksheet line by line. If you don't have it, we will send you a link to it when you get the link to the recording and the slides, and you'll have the recording so you can always go back. But the steps we work through is first we calculate how many mice we need on a weekly basis. Then we calculate the wean per female per week, and from that we can calculate the number of breeding females we need. And then we deal with our replacement schedule, how frequently we have to replace the females, and whether we actually need to factor in those additional females into the size of the colony to deal with those replacements. All right, so we start out, and I have our goals right here of what we're trying to produce, and then we just start. So we need 20 mice, so we put a 20 in the first line. Then in line 2, we deal with our age range. So if you need them all born in the same week, we put in a 1. If you need a 2-week age range, you put in a 2, 3 if you need a 3-week age range, etc. In this case, we need a 4-week age range, so we put in 4. Line 3, we deal with how frequently we need the mice. If you need them weekly, put in a 1. If you need them every other week, put in a 2. If you need them every 3 weeks, you put in a 3. Every 4 weeks, you put in a 4, etc. We want them every 4 weeks, so we put in a 4. And then what we do here in line 4 is we're going to divide line 1 by whichever is smaller, line 2 or line 3. The purpose of this is to determine whether the age range of mice or the frequency in which we need them is dictating and driving how large a colony we need. In this case, they're the same, so it doesn't matter which one I use, and so I'll get 5 for line 4. Line 5, we deal with the sex of the mice. If we want both sexes, we use 1. If we only need one sex, we enter 2, because if we only need female mice, we're still going to produce male mice, and we have to factor that into the size of our colony. Line 6, we deal with the breeding scheme, and really the way to think of this is what percentage of mice that you get out of whatever your breeding scheme are useful. So if you're breeding home by home, and also you want to convert whatever that percentage is to a fraction, and I'll explain why in a moment. So if you're breeding home by home and 100% of the mice are useful, that's 1 out of 1, you enter 1, which is what we have here. If you're breeding, say, het by het, and you're getting 25% homozygotes, that's 1 out of 4, you would put in a 4. But if you're breeding, say, het by home, and it's 1 out of 2, or 50%, you would put in a 2. If the breeding performance does not match expected Mendelian ratio, so let's say you're breeding het by het and you only get 20% homozygotes, that's 1 out of 5, you put in a 5. Because honestly, what we need here is we're just factoring in the additional size. So if we only produce 1 out of 4 mice that are useful, we need 4 times as many mice. And that's what we're doing here. And line 7, we deal with using fewer mice or not. So right now, what we're trying to do is to generate 20 mice every month. But this is an average, on average, 20 mice a month. Some months we may get 18 or 19 mice. Sometimes we may get 21, 22 mice. The question is, can you use less than 20? If you can, you put in a 1. If you can't, you need to enter some sort of multiplication factor, such as like a 1.1 to give you 10% overage, like 22 mice, or 1.2, etc. Some of you may already factor that into this number ahead of time, and if that's fine, then you use the 1. In this example, we're okay using less than 20 mice, so we just put in a 1. And now we calculate the number of mice we need weekly by multiplying line 4, 5, 6, and 7 together. 5 times 1 times 1 times 1, 5. So we need to produce 5 mice every week. And it's nice to just kind of check how this all makes sense. So if you produce 5 mice a week, every 4 weeks, we're going to produce 20 mice, which is what we need. Now we move on to calculating the wean per female per week. That's our breeding performance statistic. So these are the numbers that I gave. So line 9, that's the average number of pups weaned per litter is 5. Average number of litters per female is 2.8. And again, when you calculate this, you must include your non-productive females so that you don't overestimate the breeding performance of your strain. And then line 11, that's your breeding rotation. That's how long it takes from when you set up your matings to when you retire the breeders. We'll use 26 weeks. And we calculate the wean per female per week by dividing the average number of litters per female by the breeding rotation. And then we multiply that by the average number of pups per litter, and we'll round to the nearest hundredth, which gives us then a wean per female per week, which we're going to use of 0.54. What that means is that every female in our colony on average produces 0.54 pups weaned every week. Once we know that, and we now know how many mice we need a week, we can figure out how many breeding females we need in our colony. And again, I just put this slide back in here to, again, if you don't have good data on the breeding performance of your colony, you can use these ranges for these backgrounds to give you a place to start until you have collected the appropriate data. And again, as I said, I went with the low end of the black 6 range. So we calculate the number of breeding females then in line 13 by dividing the number of mice pups we need every week by our breeding performance, the wean per female per week. And then we round up to the nearest whole number. That'll give us 10. And you can always come back and recalculate. So if we have 10 females multiplied by the breeding performance, that's going to be 5.4 pups per week. And I need at least 5 a week. And so that works out. But now we have to think about what happens when those breeders get old. How do we handle that in the colony? So first we need to figure out how frequently we need to replace the breeding females. And it's really best to replace females as close to weekly as possible. That will give you the most consistent breeding performance. So we do that by taking the number of females we have and dividing by our breeding rotation of 26 weeks, and then we round up to the nearest half. So in this case, we're going to have to replace half a breeding female every week. We obviously can't do that, so we're looking at replacing 1 breeding female every other week. And the last part we have to think about then is, do we actually have to factor in additional breeders into the colony to deal with these replacement females that we need every other week? And you need to then look at these criteria. So if your experimental mice and your breeders have different genotypes, for example, you're breeding het by het and you're using homozygotes, or you're breeding het by home and you're just using only one sex in a couple of these scenarios, or if your age range in weeks is less than your cohort frequency in weeks. So if you need, say, in 1 week age range, but you need them every 4 weeks. If either of these apply, you don't have to factor in additional breeders. You will need to factor in additional breeders if your experimental mice and breeders have the same genotype. So you're breeding home by home, and you're using homozygotes, for example. And so you have to match both of these. Your age range in weeks is greater or equal to your cohort frequency in weeks. So a 4-week age range, say, needed every 4 weeks. If you meet both of those criteria, you have to factor in additional breeders. We actually meet both of those in this scenario, so we will need to include extra breeders into this colony. So we do that with lines 15 and 16. So what we do in 15 is we first figure out how many additional females that we need. So we take what we have here in 14, and we multiply it by 2, because every female that we need, we also have to have a matching male. And then we divide that by our breeding performance and round up to the nearest whole number. So we do that and we get 2. So we actually have to have 2 females added to this colony to then deal with the replacement breeder females that we need. And then we add that to our line 13 to give us 12 total females in this colony. And just to see how this then works, so if we have 12 females times our breeding performance, we're going to produce about 6.5 pups every single week. And so what we're going to do is every single week, we're going to hold all 6 of those pups. It's 2 boxes, one for the males, one for the females. And then every other week, we're going to replace our oldest breeder with a new pair. Right? So 6 pups, we have 5 for experiments. 1 is our 1 replacement breeder. That's essentially what we've got going on here. And then from that, we can actually figure out how much box space we need to produce every one of those cohorts. If we breed in pairs, we're going to need 12 boxes just for breeding. Breed in trios, we'll need 6 boxes. We're going to need 10 boxes for holding all of the mice because we need 2 boxes, 1 male, 1 female every week from 3 to 8 weeks of age, so that's 5 weeks. And then total, that's 22 total boxes if we breed in pairs, 16 total boxes if we breed in trios. Now this only includes the mice, the box space you need to actually produce your cohort. You're going to have to factor in additional space to actually do your experiment and for your controls. This is only for your experimental mice. And so now you can look at, do you have enough space, and make sure you have that space available to do the work that you need done. And we can start to think about how much is this actually going to cost to produce every cohort. So, and I know people look at their per diems in different ways, but basically if you breed this in pairs, for each cohort, you're going to have 76 box or pen weeks. So you have 12 boxes that you have on the week shelves for 4 weeks for each cohort. That's 48 weeks. And then the holding for your experiments and replacement breeders is 28 weeks because you have 2 boxes that you hold for 10 weeks or 5 weeks, 2 boxes that you hold for 4 weeks, 2 for 3, etc. So that's a total of 76 box per week. And if just using an example per diem, you could substitute in your own of 75 cents per box per day or $5.25 per box per week. That's about $400 for the cohort or $20 per mouse. If you breed in trios, it's going to be a little bit less because we only have 6 breeding boxes, so 52 weeks total box weeks. And again, with the same per diem, $275 for that cohort or $13.75 per mouse. And so you can use this to potentially estimate what it's going to cost you to produce every cohort of those mice. Okay, now that we've gotten through that, what I want to do is now go through with looking at these parameters and these research needs, the age range, the sex, the number of mice, and the breeding scheme, and see how they can affect the colony size for this strain so that you can see what those decisions you make, how they're going to affect your colony. So here's our original scenario, 20 mixed-sex homozygotes, 4-week age range every 4 weeks. Let's change that to a 1-week age range. So we need them all at 8 weeks of age. And let's go back and look at what changes that makes in terms of all the worksheet and then how large that colony will be. So what I have here in the blue, the left-hand column is the original scenario, and in the red in the right-hand column is our new scenario. So we still need the same number of mice, but we're going to adjust line 2 because we have to put in a 1 because we need a 1-week age range. So they all have to be born in the same week. That's also going to adjust line 4 because we have to divide line 1 by the smaller of lines 2 and 3. So we divide 20 by 1. That will give us 20. None of these change at all. But what will change is line 8, the number of mice we need weekly. So we're just multiplying lines 4, 5, 6, and 7. And that gives us 20. So now we actually have to produce 20 mice every single week, when previously we only needed 5. The breeding performance is the same, and so I'm not actually going to show it again for these different scenarios. What I'm going to do is I'm actually just going to start showing only the stuff that changes so that we don't have to go through every single line. So I'm not going to show this again because we're going to be using the same exact data for every single scenario. We will have to change the number of breeding females. So again, we divide the number of mice we need every week, which is 20, by our wean per female per week. And now we need 38 females on the shelf. And again, I can make sure this all works. 38 females times our breeding performance, 20.5 pups every single week. So that works out right. Next, we need to look at the replacement females I need every week. So I take that 38 females, divide it by my breeding rotation of 26 weeks, round up to the nearest half. That's 1.5. Again, I can't replace half a female every week or at all. So I'm looking at replacing 3 breeding females every 2 weeks. So I can either do it every 2 weeks, I replace 3 females, or I could say replace 1 female and the next week I replace 2, then 1, then 2. Whatever is most convenient for how you would maintain your colony. But now do we have to factor them into the size of our colony? Remember back here, if our age range in weeks is less than our cohort frequency, we don't have to factor in additional breeders. So in this case, we won't. And you'll see that we won't have to do that and how that works out. But we don't have to factor that in. So therefore, our colony is there at 38. So 38 females times our breeding performance, 20.5 pups per week. So what we're going to do here is the first week we're going to hold all 20 of those pups, 2 to 4 boxes. Those are our experimental mice that we need. And then the next week we're going to hold enough mice to replace our 6 pairs or 3 trios, depending how we breed, and then we discard the rest. And then in the next couple weeks, we discard all of the pups that we produced. So if you look at it, in this scenario, we're discarding more than half of the mice that we produce because we just don't need them for the experiment, which is potentially a waste of resources. So is there a way maybe you can make adjustments? If this is a common inbred strain like C57 Black 6J and you absolutely need a 1-week age range, maybe it's just best to buy the cohorts when you need them and not even worry about having to produce them, especially if the cost of purchasing them is very close to the cost of actually producing them yourself. And then again, we're going to replace the oldest breeders this week, 2 the next week, 1, 2, we're just going to alternate that to rotate our breeders on a regular rotation. We can figure out how much box space again we need. So if we're breeding in pairs, 38 boxes, 19 if we're breeding in trios, and we'll need 10 boxes for holding all of those mice that we need. So 48 total boxes for pairs, 29 for trios, and again, you can make sure that you have the appropriate space. And again, we can calculate the cost of those mice, and we're going to use the same per diem cost of 75 cents per box per day. If we breed in pairs, that's 178 box or pen weeks, so 38 breeding boxes, 4 weeks per cohort, 152 weeks. And then the holding for the experiments and replacement, we're going to hold 4 boxes for 5 weeks. That's 20 boxes, that's our cohort. And then 2 boxes we hold for 3 weeks. That's the replacement breeders, so a total of 26. So that's 178 total, 935 for that cohort, or 46.75 for that mouse, per mouse. And again, I can do the same thing with trios. It'll just be a little bit less because we only need 19 boxes, so there it's 26.80 a mouse. And again, if this cost is getting close to what it would cost to purchase the mouse commercially, then maybe it's just easier to buy them and not have to discard as many mice. And I'm not going to go through this pricing for the rest of the scenarios. I think I kind of outlined how you would figure that out, just so we're not going to do that for everyone. I got that information to you. So we're just going to keep going through examples, but I'm not going to go through the pricing portion again. Okay, so we'll come back to our original scenario, 20 mixed-sex homozygotes at a 4-week age range every 4 weeks. And let's switch it to single sex. So we only want females in this case. And let's work through how that would adjust our colony. So I'm only going to show the lines that change in this. So I'm going to skip all the other parts of it so that we can get through this just a little bit more efficiently. So what we're going to adjust here is line 5. So we're going to put in a 2 because we only need 1 sex. And that's going to then adjust line 8, which is the number of mice we need per week. So we multiply 4, 5, 6, and 7. 5 times 2 times 1 times 1. So 10. So we need to produce 10 mice every week. 4-week period, that's 40 mice. But again, that's going to be 20 females, which we need, and then the 20 males, which we only need for breeding. Determine the number of breeding females that we would need. We divide the total number of mice we need on a weekly basis by our breeding performance. And that gives us 19 and round up to the nearest whole number, and that gives us 19 total females. And again, I can work through the math on that just to make sure that that worked out correctly. So I take that 19, divide it by our breeding rotation of 26 weeks, round up to the nearest half, I get 1. So for this colony, I would want to replace 1 breeding female every single week. Now we have to determine if we need to factor in replacement breeders. So we're breeding, the experimental breeders have the same genotype, and our age range in weeks is equal to or greater our cohort frequency. Both of those, we match both of those criteria, so we will need extra breeders in this colony. We'll have to factor them in. So we again take this 1, we multiply it by 2, and then divide it by our breeding performance. That gives us 4. And so then we add that to the 19, and we need 23 total breeders in this colony to produce the experimental mice that we need and our replacement breeders. The way this colony then would look is we'd be producing 12 pups every week, so that's about 6 pups every week. So basically every week we're going to hold all 6 of those females. And then in 1 week, we need to hold about 4 of those males for breeding purposes. May depend on whether you breed in pairs or trios, but and you may take a couple extra, but that won't require an extra box. And then every week, we're going to replace our oldest breeding females. But what you're going to see again is that most of the males that we produce in this colony, we're going to discard, which again means we're discarding a lot of the mice that we produce, which basically means you would be using resources to produce mice that you can't use. Again, you could look at making adjustments. Do you really need to use only one sex, or maybe could you use both sexes in your experiments? Or again, if it is a common strain like a C57 Black 6J, and you absolutely only need female mice, you could potentially consider looking at just purchasing the females so that you're not actually producing males that are not going to be used for experiments. And we can take this again and calculate the box space we need. So we need 23 boxes if we're breeding in pairs, 12 for trios, 6 boxes for all of the holding, and we can figure out our totals. And again, make sure that you have enough space to produce those mice that you need. I want to look at now changing the total number of mice we need, because this brings up a really good point I'd like to discuss. So we're going to jump from 20 mixed-sex homozygotes to 100 mixed-sex homozygotes. And before we get into that, I want you guys all to think about, and if Sarah, if you can push out that polling question, we're basically increasing the 5 times as many mice as we want. So how are we going to actually size that colony? Are we just going to use 5 times as many female breeders as we used before? Pretty simple. Or do we need to calculate the total number of breeding females using that worksheet? And we'll give you 30 seconds, a minute or so to answer. It's whatever Sarah feels is right. And then she'll post it, and we can all see what people answered. All right. Looks like she posted. Looks like by far most of you went with B, that we have to calculate the number of breeding females using the worksheet. A few of them thought we just use 5 times. I'm not going to actually give you the answer right now. I'm going to walk through this and let's see what happens when we do calculate that using the worksheet. And we'll figure out the answer at that point. But I think most of you probably know. Okay, so in this case, we're changing line 1. We're jumping up to 100 mice from 20. And we also adjust how many mice in line 4. So again, the math, we just now need 25 there. Everything else is the same until we get to line 8, where we calculate the number of mice we need per week. Again, we just do this 4, 5, 6, and 7, multiply together, 25 mice per week. That is pretty straightforward. But let's look at the breeding females that we need. So we take that 25, divide by our breeding performance, and we get 47. And I'm going to hold off on commenting on about how we got there until we get all the way through this. So we would need 47 females times our breeding performance gives us a little over 25 pups per week. So we know that worked out right. But let's look at our replacement females. So we take that 47, divide it by 26, round up to the nearest half. That would give us 2. So we're looking at replacing 2 females every week. We're going to have to factor them in because we're breeding home by home to get homes. They have the same genotype. And our breeding range and age is equal to our cohort frequency in weeks. So we then come back, so we take that 2, multiply it again by 2, divide by our breeding performance. It gives us 8 total females that we need to just deal with the replacements. We add those 2 lines together, gives us 55 total breeders. So just for those of you who looked at this, if we go back, previous scenario, we needed 12. If we multiplied that by 5, that would have given us 60 total breeders, but we really only need 55. The reason for that is, remember, through part of this, we are doing some rounding up or rounding. Yeah, we are doing a little bit of rounding, which is why it's not a sort of linear relationship between the total number of breeders and all of this. So basically, every time you make a change, even if it's doubling the number of mice that you need, or 5 times as many mice, or 10 times as many mice, you have to go through the worksheet again to calculate that number. Because of that, the rounding that we do, it doesn't work exactly that way. So if we had just taken that 12 and multiply by 5, we'd have had 60 breeders on the shelf, which is 5 more than we needed, and that would have been a larger colony, and we would have been wasting more resources until we figured out that our colony was producing more than we needed. So again, every time you make a change, you have to recalculate. But that was really great that most of you got that question right. That's really good. So the way this colony then would look is 55 females times our wean per female per week. That would produce about 29.7 pups per week. Basically, every week we're holding all of the pups, and then every week we're replacing 2 of our oldest breeders. And again, we can see how large that colony would be. 85 total boxes if we're breeding in pairs, 58 if we're breeding in trios. And the last example I'll go through is changing the breeding scheme, just to walk through that. So we're going to switch from breeding home by home to a breeding het by het. Maybe it's because your mouse strain is on a mixed genetic background and you need to use colony controls. Maybe the homozygotes of your knockout are infertile and you have to breed hets. Or maybe the area of research or the journals that you publish in require colony controls. Whatever that reason, let's just see what happens when we do this. And then we need 20 mixed sex homozygotes, 4-week age range every 4 weeks. So what we're going to change then is line 6. That's the breeding scheme. We're going to assume in this scenario that by breeding het by het, we produce 25% homozygotes, or 1 out of 4, and we enter a 4 into line 6. And then for line 8, the number of mice we need per week, we factor that in. So now we need to produce 20 mice per week, which is basically 80 mice every 4 weeks. But remember, only 1/4 of them are useful, which means that we get 20 useful mice that are homozygotes, which is what we're looking to get. In terms of the number of breeding females, we divide that 20 by our wean per female per week, round up to the nearest whole number, and we get 38 total females. And again, we can walk through that and make sure that that makes sense. That's going to give us 20 plus pups every week, which is what we need. In terms of our replacement schedule, we divide that 38 number of females by the breeding rotation of 26 weeks, and that gives us 1.5 females every week. And again, we can't do the half female, so again, 3 females every 2 weeks, or you alternate 1 female and 2 females, 1 female, 2 females, etc. Now in this case, we're breeding het by het and using homozygotes, so we do not need to factor in additional breeders into this colony. So that's where we stay at that 38. And the way this would look then is if we have 38 females, 20 pups per week, we're going to get approximately 5 homozygotes that we hold for our experiments, 10 heterozygotes that we use some for replacement breeders, the rest that we don't need, we just discard, and then the 5 wild type, which we now can hold for our controls. Now in this last scenario, this colony is producing both our experimental mice and our control mice. So the other scenarios, the controls were separate. So just make sure that if you're doing this, this is actually including your controls as well. And so basically every week we're going to hold all the homozygotes, all the wild type mice, and then we'll hold enough of the heterozygotes to produce our replacement trio, pairs or trios for that 4-week range. And then we replace our oldest, 2 oldest, oldest, 2 oldest, and we just keep going through that. And again, we can actually figure out how many boxes, so 38 pairs or 19 trios, 22 boxes for all of the holding, 60 total if we breed pairs, 41 if trios. And again, in this scenario, the controls are included, but this gets you just up to the cohort. You'll still need the space for the actual experiments. Okay, I know that was a lot to go through, but we're just going to finish up with just a little bit more information. So when you're thinking about breeding and your total cost of what it costs for you to produce the mice you need for your experiments, there's the monetary cost. There's the per diems. Realize that, of course, if they are supplemented by your institution, their overhead from your grants may be funding that. There's the salary of the technicians to maintain your mice, cost for genotyping the mice. But just realize that there are some costs that are not monetary. There's the time that you use to manage your colonies, time you need to collect and analyze your breeding data, the time you need to plan out your colony size so it's sized accurately for your experiments. And there's also the space that uses, that you're just using for breeding mice, not for experiments. And so if there is something that you can think about. And if the cost in terms of monetary is getting really close to the cost of just buying a mouse, you can save yourself time and space of breeding mice, buy them when you need them, and that will free up that time and space where you can then do more experiments and actually potentially get your work done a little faster and move your research program along faster by not having to manage mice yourself. Just some general colony management tips. And if this is something of interest to you, I did a whole webinar 2 weeks ago on colony management, more efficient colony management. If that's of interest, you can just email us at the email address at the end and ask for that, and we can send you the recording. But mate your mice early, by 4 to 12 weeks of age. Choose your breeders carefully so that you avoid any sort of selection pressure. You may think that if this one breeding pair breeds better than average in your colony, using their pups as breeders might be advantageous to improve the breeding performance of your colony. It may do that, but you may accidentally select for something else or select against the trait of the phenotype of your colony. So it's best to really just randomly select your breeders. We've talked about replacing your breeders on a regular rotation, whether it's weekly or monthly. The closer you can get to weekly, the better, depending on the size of your colony. You want to replace those non-productive breeders as soon as possible. Most strains, we give them 60 days to successfully wean a litter. Unless we know they're slower, we might give them 90. And again, I can't emphasize enough how important it is to collect your own breeding statistics and evaluate them regularly so that you can accurately size your colonies. Some other factors that can impact the consistency of your breeding performance, good pedigree and lineage tracking, so you know which mice were the parents of which other mice and you know how they're related, can help you that with lineage tracking. So if any spontaneous mutation arises in your colony that gives some sort of unusual phenotype that you want out, if you know how they're related, you can get that mutation out of your colony easier. Any pathogens that mice carry can impact breeding performance. So you want to look at your health monitoring program and your barrier and how comprehensive it is to make sure that your mice are as healthy as possible. And then a really good quality diet with really good quality control on ensuring the consistency of the food that your animals are eating. Sometimes different grain products can impact the breeding performance and weight. So variation in the diet can lead to variation in weight. And we have found that the weight of the breeders can impact their breeding performance. So having a diet that is consistent will help keep your breeding more consistent. And then your technician and training and experience. The more experienced your mouse room technicians are, the better they're going to be able to see that there's observe problems that may occur in a colony, whether it's a spontaneous phenotype or something like that, earlier and help you deal with it. The earlier you find these problems, the better. So make sure that that they get good training and that you really listen to what they have to tell you. And then really consistently following your standard operating procedures, your SOPs, ensures that everything in the mouse room remains as consistent as possible so that your breeding performance remains as consistent as possible. Because the goal really here is consistent breeding performance so that your colony is consistent and you know how many mice you're going to get how often. All right, so that kind of wraps things up. So we've really talked about good breeding performance data collection, your research needs, how to calculate breeding females for a steady state colony, and then how things like the age range, the sexes, et cetera, impact your colony size and how you could potentially adjust them to meet your existing resources. So I just want you to know, the Jackson Laboratory, we do offer a whole host of services that might be helpful to you in cryopreservation, breeding. We can generate new, if there's strains that you're thinking of crossing like a Crelox, but you're not able to get into your facility, we are functional and we could start that breeding for you. And just think on, with most of us being working from home and dealing with this pandemic, think about how you can plan and rebuild your research, your colonies, reprioritize how you work so that you can prevent those delays later. If there was a strain you had us cryopreserve or a strain that we have cryopreserved that you're going to want when you get back in, now's the time to start thinking about having that strain cryo recovered and then even potentially bred to expand so that when you walk back into the lab, your colonies are ready and you don't lose that time waiting for those mice. Again, remember, about 3 months every generation for breeding. If there are mice that you know you're going to need when you get back, even if you don't know when you're going to be able to get back in your lab, you can pre-order those mice. We are taking orders without a ship date, and we'll hold that until you know that and you let us know. But it helps us gauge the demand for the colony so that we can have those mice available when you get back. We are doing like in vivo studies. So if you're looking at doing in vivo efficacy studies on a disease model, we are doing studies. We are open. We can be collecting data while you're not able to be in the lab right now and help keep your research on track. So don't lose the time that you have. Look at coming to us and how we maybe help you keep your research program going, even if you're not able to get into the lab.
I'm so sorry, I lost track of with all the questions. Thank you so much, Peter. Can you hear me?
Yes, I can. Thank you, Sarah.
All right, wonderful. And that is a great talk. It's there's so much going on, so definitely want to let Peter get a glass of water, something to drink. Take a deep breath. I'm going to tell you about a couple upcoming JAX webinars. So over the next month or so, we've got a couple interesting webinars coming up. So on May 28th, we're going to have a webinar on therapeutic antibody drug discovery using humanized FcRn mice. Then in June, we're going to give a webinar comparing different immunodeficient mouse models for cancer, immunity, and transplant research. In the middle of the month, we're going to have how to generate better mouse models for your research, so different approaches and considerations to consider. And then later, we'll have a webinar on how many are too many, so about mouse caging configuration and research studies. And the other thing we want to discuss is, so we've been working together as a group to try and figure out more ways that we can better support the research community. So as the technical information team has started these live 15-minute chats with our JAX TIS scientists. So we're going to be discussing a couple different topics. We've cryo-recovery, cohort size, information about gang cohorts, and the next one up is going to be on talking about how to generate new colonies. And we are now in our live Q&A, so we got a lot of questions. They came fast and furious. We're still getting some at the end. We have tried to answer as many questions as possible during this Q&A session with my wonderful colleagues, Xing Tong, Elizabeth, and Caitlin. And any questions that we didn't get to, please send them to mytech@jax.org. We're always happy to help assist you with your research needs. And I also want to mention at the end of this webinar, when you close out of it, you'll get a brief survey. Please do us a favor and fill that out. It really helps us with generating either new webinars or even just helping us to better address our webinars to your research needs. All right, Pete, so we got a lot of questions. We tried to answer as many as we could, but I've got a couple for you that I think would be really great topics to consider.
Okay,
so
go for it.
One that we got a lot of questions on, actually, was the difference between breeding by pairs and trios. And I wonder if you could comment on kind of the benefits of each method.
So I'll just lay out the basics just in case anybody isn't sure. A pair would just be one male, one female in a box for breeding. A trio would be one male and two females in a box for breeding. The trios, one advantage of trios is you essentially have two breeding females in one box, so the number of boxes you have on the shelf is halved. So it just takes up less space on your shelves, which means it uses less resources for you. You may want to consider looking at, how well your mice breed in pairs versus trios to see if there's a difference in their breeding performance. But again, and some facilities won't let you breed in trios, that you can't have as many mice in a cage as that. But you may want to consider that webinar if that's a situation you find, and this may be a webinar to attend because we have looked at the impact of pairs versus trios in different size cages to look at breeding performance. And there is some good data in that talk about that. but so you have to go with what your facility regulations are, right? So if they don't let you
Mhm.
breed in trios, you can't breed in trios. But if you can breed in trios and it doesn't impact the breeding performance significantly, it just uses less resources.
Okay, great. We also got a couple questions that were fairly similar, and so I'm going to summarize them kind of all together, which was related to the difference between needing experimental groups of different ages. So you want to generate mice, and you're looking at 3 months, 18 months, 12 months, kind of a variety of ages. Or a corollary to that was, what do you do for when kind of the phenotype manifests later in development? So they seem sort of similar, so I wanted to get your impression on that concept of different ages that you're studying.
Yeah. So, um, well, you start taking the time to plan this out is going to help you work through that.
Mm-hmm.
If you need, part of it is, if you need a cohort of mice at 3 months of age and a cohort of mice at 6 months of age and one at 12 months of age, et cetera, are they all the same size, right? That's the first thing. If they're all the same size,
Mm-hmm.
um, you're looking at basically producing 20 mice, but you need them every 3 months, right? So that's every 12 weeks, and you just walk through it, right? You may hold them longer. Some you may
Mm-hmm.
hold them only to 12 weeks of age. Some you're going to go to 26 weeks of age. Some of them, whatever, a year of age. But it's how frequently you need them, right? And so if I need 5 cohorts of mice that are all 3 months apart in age, I just walk through this, and that's my frequency is every 3 months. Um,
Mm-hmm.
if you know that your phenotype, say, is let's say only 80% of your mice that you produce are going to have the phenotype you want. That's where you either factor it in with the number of mice you need. So if I need 20 mice that have the phenotype, but only 80% of the mice have the phenotype, I just have to up the number of mice accordingly in terms of my cohort size that I need so that I get the numbers of mice that are going to be useful. Does that make sense?
Right. Yeah, yeah. So sort of similar, like if you have, so another question on that regards is, what if you have a phenotype that has an early onset, and how will that affect? So you have a phenotype that may affect breeding and has an earlier onset. What would you?
Yeah, so I guess it's going to depend on how early the onset is, and is it a specific genotype? So, for example, if your homozygous mutants have a disease phenotype that comes on very young, and those don't breed, you may have to breed head by head. Or say it's a phenotype that comes on like 4 months of age, and the male homozygotes will breed, but the females won't. You breed het females to home males, but your breeding rotation
Mm-hmm.
may be shorter, right? So then you would have, when you come to calculating the wean per female per week, part of this is factoring in your breeding rotation. So if you only keep your mice breeding for 10 weeks and that's all you got, that's all you got. And of course, it's going to probably mean you have a larger colony to produce the same numbers of mice, but that's how you would factor that in. So you can, it depends on the phenotype. I mean, if it affects one sex versus the other, you can manipulate your breeding scheme to deal with that. You also may have to shorten your breeding rotation. And again, but that's going to be part of the data that you've collected. I mean, if they only throw 2 litters, and you have to retire them at, you know, 20 weeks of age, you have to retire them that young, and that's all you get.
Okay.
But you can manage.
Yeah. All right. I don't want to keep everyone too much longer. I think it's been a great talk. I think it's always very interesting to think about the ways we can plan in advance, in terms of generating the numbers that you need. The advanced planning is always beneficial. Do you have any last comments for our listeners today?
I don't. Again, really the most important thing is collect good data. The better the data you have, and evaluating it regularly will help you size your colonies as efficiently as possible. That's the biggest thing, just watching that data.
All right. Well, I want to thank everyone for attending and for participating in our Q&A session. And again, any final questions, any questions you come up in the future, please email that MiceTech email, and we're always happy to help you. All right, and I'll say goodbye.
All right. Thanks, everybody.